Okay, great. And we are live. Thank you everyone for joining us at the MSU Science Festival's Science After Dark. I'm Katherine Hagman from the Science Festival. Today I'm also joined by Roxanne Troon from the Science Festival. And today we're really excited to welcome Dr. David Morgan, from the director of the MSU Alzheimer's Alliance. Welcome.
Thank you.
So before we dive into today's topic, would you mind just telling us a little bit about your background and what you study, and a little bit of an introduction to what we'll be talking about tonight?
Yeah, well, I'm a research neuroscientist. I've been working now on Alzheimer's disease for about 35 years. I did my undergraduate and graduate work at Northwestern in Chicago, and I spent a decade in Los Angeles at the University of Southern California. And then I spent 25 years in Tampa, Florida at the University of South Florida, where I became the CEO/director of a multidisciplinary research center dedicated to Alzheimer's disease called the Bird Alzheimer's Institute. And I moved to Grand Rapids about 3 years ago, right near the end of 2017. And here we've been developing both clinical and basic science research programs, again, focused on Alzheimer's and Alzheimer's research. For 5 years, I was on the program committee of the Alzheimer's Association International Conference, which is a 5,000-person conference that focuses on Alzheimer's disease. And so I'm fairly broadly connected with this disorder and trying to understand how we can have some impact upon it.
Great. Well, I'm excited to hear more about your talk, so we can go ahead and dive right in if you're ready.
All right. So what I'm going to talk about today is a little bit of the history of our scientific understanding of Alzheimer's disease. And I'm going to talk a little bit about some of the newer medications that are coming out that seem to be having some meaningful impact on the progression of the disease. And then I'll kind of give you my take on where we're going to be in the next 5 to 10 years or so. We've started a center in the College of Human Medicine called the Alzheimer's Alliance. And while it's centered at Grand Rapids, there are members also on the Lansing campus as well. And a number of people outside of MSU are also participating in this Alzheimer's Alliance. And in particular, the healthcare systems here in the Grand Rapids area are active in this as well. We also are partnering with an organization called Rethinking Dementia. And this organization consists of largely the residential care facilities that take care of people in memory care centers in trying to find ways to support the needs of their patients as well as the families of those patients. We're interested in laboratory research, and there's about half a dozen scientists here in Grand Rapids studying in a laboratory setting. We want to develop clinical trials to prevent and treat dementia. Grand Rapids is the 50th largest metropolitan area in the United States and has about 1 million people in the greater Grand Rapids area. And if you look up Alzheimer's and Grand Rapids in clinicaltrials.gov, there is nothing. And so we're trying to see if we can expand that. And then we also want to try and facilitate early diagnosis, treatment, and enrollment in clinical studies. It turns out only about 1/3 of people with memory disorders ever get a medical diagnosis. And this is the new research building that we're in here in Grand Rapids. Alois Alzheimer was a German physician. He was a neurologist and a pathologist, and he based his original description on a single case. This was a woman whose name was August Dieter. It turns out I can tell you August's name without violating HIPAA, because HIPAA no longer applies after you have deceased, interestingly enough. She started to exhibit a dementia syndrome, importantly, at a relatively early age. She was in her early 50s, and she started to have this disorder. Now, had August been over the age of 65, it's likely Alzheimer never would have bothered to look at her brain. And that's because at that time, and really until fairly recently, it was felt that all of us, if we lived long enough, we would end up with some kind of cognitive disturbance associated with aging. Sometimes they called it senility, sometimes they called it hardening of the arteries, but whatever they called it, they didn't think of it as a disease. But because August was so young, Alzheimer took her brain and he sectioned it after she died, and he stained it with what at that time was a high technology, which was silver chemistry. And the silver chemistry was transferred over from the photographic industry. So in some sense, when he looked at her brain, he had essentially developed it like you would a photograph. And when he did this, the silver bound to these rather unusual structures that he found in her brain. And one of these is outside of neurons. This is called a senile or a neuritic plaque. And then he found inside neurons these twisted fibers, these filaments that were located inside the neurons. So these extracellular deposits we call amyloid plaques. The intracellular deposits we call neurofibrillary tangles. And amyloid plaques are made up out of a protein called the amyloid protein, or A beta. And neurofibrillary tangles are made up out of a protein we call tau, T-A-U. And to this day, if someone dies with dementia and you examine their brain and it has plaques and tangles, that is the definition of Alzheimer's dementia. Now, for many years, Alzheimer's only applied to people under the age of 65. It was called pre-senile dementia because senile dementia wasn't a disease. But in the 1980s, pathologists noticed that many people in their 80s and 90s dying with dementia had this plaque and tangle pathology. So when I first got into this field, it was called senile dementia of the Alzheimer type. Finally, around 1990, we just decided to drop the specification, and Alzheimer's disease now applies irrespective of age to anyone who has this plaque and tangle type of pathology. So what do you think the biggest risk factor for Alzheimer's is? Well, it's not cigarette smoking and it's not lack of exercise, although both of those can contribute. It's really just too many birthdays. Age is without question the greatest risk factor for Alzheimer's disease. And even though my wife complains that she isn't having any more birthdays, I think she's still increasing in her risk every year. The disease, while not quite at the pandemic level, is still reaching epidemic levels in the United States. And part of the reason is people are simply living into the age of risk of developing Alzheimer's. We've reduced heart disease by 50% on an age-adjusted basis over the last 50 years, and we're starting to make significant inroads in cancer, and people are living into the age when they start to develop this disease. Interestingly, the average age of death of an Alzheimer patient is older than the average age of death in the general population. But we now have, and this is slide's a little old, about 5.8 million people in the United States are estimated to have Alzheimer's or another dementia. We estimate that as the baby boom generation retires and we continue to see gains in longevity, this is going to reach 16 to 20 million people by the midpoint of this century. Today, 1 in 10 people over 65 years have some form of cognitive impairment. Over 85, it's probably 40%. And what this means is that in the state of Michigan, 1 in every 50 people have some form of cognitive impairment or dementia. And again, only about a third of those ever receive a formal medical diagnosis. It's increasing dramatically as a cause of death, and it's really deceptive to say that it's the 6th leading cause of death. And the reason is the following. We have about 600,000 people a year who die from heart disease. That's number one. We have 570,000 who die from cancer. There are 500,000 people annually who die with severe dementia. Now, it doesn't show up on the death certificate as the cause of death because it doesn't directly cause it. But there's almost as many people dying with severe dementia as a contributing factor as are dying from heart disease and cancer. Estimated medical care costs. This is not soft costs like lost productivity. Medical care costs are $250 billion in the United States. This is slightly more than heart disease and about 30% more than the medical care costs for cancer. If we could delay the rate at which someone goes into residential care by 5 years, we save half of that cost. This $250 billion is 1.2% of the gross domestic product of the United States. This is a massive, massive medical problem that we've really not until recently started doing anything about it. About 5 years ago, we spent $6 billion on cancer and $600,000 on Alzheimer's disease. I and a number of my colleagues have gone to Capitol Hill on multiple occasions, and we have been talking to the senators and the representatives to try and convince them of the significance of this problem and how much it's ultimately going to cost the country. Interestingly, we've been successful. We now have a budget of almost $3 billion for Alzheimer's disease research. Over the last 5 years, we have essentially quintupled the amounts of funds available to study this disease and find cures and treatments. So I think that, we're really having an impact when we do go in and talk to these representatives and senators. Now, you'll often hear people like myself talk about dementia and talk about Alzheimer's and talk about them in kind of the same breath. And in reality, dementia is a much broader category. Dementia is any loss of cognitive function associated with age that is secondary to damage occurring in the brain. So you have to show some form of atrophy or damage to the brain tissue. When we look at autopsy, about 2/3 of the time, we find the plaque and tangle pathology that Alzheimer described. Probably the second major cause of dementia are what we call a vascular dementia. It's a series of small strokes, and these small strokes in and of themselves don't cause any overt symptoms. But as they cause increasing amounts of damage to brain tissue, they have a gradual decrease in cognitive function that develops. Many people have multiple pathologies in their brains. And in fact, increasingly, as we look at more and more pathologies, while somebody may be called Alzheimer's disease, they could have a little bit of Lewy body disease or a little bit of the frontal temporal lobe types of problems. And there are these other types of conditions that can cause dementia. But here's a key point. Many memory disorders are reversible. They're not a true dementia. There can be, vitamin imbalances, hormone imbalances, reactions to medications that you may have been taking for some time, all of which can cause cognitive impairments that could be considered very much like a dementia and very much like Alzheimer's disease. And memory care physicians are extremely adept at discerning who has a true dementia and who has something that we may in fact be able to treat. And probably about 10 to 15% of people who come into a memory disorders clinic don't have an Alzheimer type dementia, but actually have something that could be quite treatable. So our first hypothesis that we used to develop medications to treat Alzheimer's disease, we call the cholinergic hypothesis. This has to do with a neurotransmitter called acetylcholine. Neurotransmitters are the chemicals that communicate between neurons inside your brain, and they do this at their junctions that we call synapses. And one neuron releases the neurotransmitter, and the second neuron detects and responds to that neurotransmitter at the synapse. And so if you look here, these are where the cholinergic neurons are, and they go up into the parts of the brain we call the cortex and the hippocampus. And at the synapses between those neurons, the acetylcholine is released into the synaptic cleft. And it's shown here as these little yellow dots. And it goes across the cleft, it interacts with the receptor for acetylcholine, and it causes a response. Well, it turns out that there's something to inactivate the acetylcholine that's called an enzyme, acetylcholinesterase. And that's what these little red Pac-Man type things are located here. Well, what we found early on was that while many neurotransmitters decline in Alzheimer's, the one that declines the most and the earliest is acetylcholine. Well, we noticed in Parkinson's disease, where the neurotransmitter dopamine was the primary neurotransmitter that was depleted, if we did something to boost the activity at dopamine synapses, we were able to, in fact, reverse many of the symptoms of that disease. We didn't slow progression, but we reversed the symptoms and gave people many years of functional life back. So we tried to do this in Alzheimer's, and we developed drugs to block these Pac-Man type things called acetylcholinesterase inhibitors. And they do block the enzyme, and that increases the amount of time the acetylcholine resides in the synapse and increases the activity through these receptors on the opposite side of the synapse. So the drug Aricept, and there's 2 other similar medications, all work in this particular way. The problem is this isn't the only problem in Alzheimer's disease. And while these drugs produce a 6-month to perhaps 2-year reversal of symptoms, they're not treating the underlying problem. They're really just treating the symptoms. It's kind of like taking an aspirin to treat pneumonia. You'll feel a little bit better, but the underlying disease goes on until after a while the aspirin doesn't do anything for you. And so these drugs have some benefit. They do delay the time that a person can stay at home before they have to enter residential care. But they don't really slow the progression of the disease in any meaningful fashion. So in the 1990s, we developed a second hypothesis we call the amyloid hypothesis. Amyloid, as I'd mentioned before, forms the core of this thing we call the neuritic plaque that Alzheimer himself had described. And what the plaque is an accumulation of fibrils made up out of these little peptides. There's little pieces of a big protein aggregate together, and they form these fibrils that essentially form a core. And around the core is a zone of degenerating bits of neurons, synapses, dendrites, axons. All of these things are brought into this core. Importantly, we also find that the immune system representatives in the brain, the glial cells, they react to this core of amyloid. And so you have inflammation that's occurring in the vicinity of these neuritic plaques. We know that there's a protein that amyloid is produced from. It's called the amyloid precursor protein. And if we have mutations in that protein, it can cause Alzheimer's in a dominant fashion. That is, if you have this mutation, you are going to get Alzheimer's, and you're probably going to get it in your 40s or 50s rather than in your 80s and 90s. We've produced mouse models, and our research team was involved in production of some of the earlier mouse models. And in these mice, we find they lose their memories, but they don't show degeneration. They don't show atrophy of the brain. Their brains don't shrink like they do in Alzheimer's. An Alzheimer brain weighs about 2/3 of its normal weight at death. A third of the brain mass is lost during the 10 to 20 years that this disease occurs. Another interesting thing we've now learned is that amyloid deposits are occurring decades before the onset of symptoms. You can think about it like cholesterol is building up in your arteries before you have a heart attack. So amyloid is developing inside your brain. In the double aughts, we added another piece to this, which has to do with these neurofibrillary tangles that I talked about, that has to do with this protein called tau. So tau is normally only found in the axons of neurons, but in Alzheimer's disease, it migrates into the dendrites of the neurons, and it aggregates. There's a lot of tangles inside this one. You can just see a few of them inside this neuron right here. If we take them and look at them under the electron microscope, they have this twisted ribbon type of an appearance. They're essentially a double helix, but in this case, a double helix of protein and not nucleic acids. And they're very hard to dissolve. It took a number of years to find agents that would let us break these down enough to figure out that this tau protein was what they were made up out of. After a neuron dies, these these remain behind as a kind of a skeleton. They're called ghost tangles or tombstone tangles because they represent the leftover skeleton of that neuron. We can have mutations in tau that cause a dementia, but it's not Alzheimer's disease. It has a different set of symptoms that's called frontal temporal lobe dementia. And it can be readily discerned from Alzheimer's dementia. Essentially, the personality is more impacted than memory at the early stages of the disease in people who have the tau mutations. These tau mutations do not cause the accumulation of amyloid in the brain. They just cause the accumulation of these neurofibrillary tangles. And so now we're starting to integrate all of this stuff together. And one of the more recent identifications that we've had genetically is to show that genes associated with this innate immune response to, my goodness, it started to rain here. Sorry. Genes that are associated with the inflammatory response in the brain have mutations that can also cause Alzheimer's disease. And this was very surprising, but what it tells us is that the inflammatory response is somehow participating in the pathogenesis of the disease. So it's not just amyloid accumulation and it's not just tau pathology, but the inflammation appears to be another important step in the process. And we think that amyloid's gradually building up over time and in and of itself is relatively innocuous. At some point, the innate immune system reacts to it and produces an inflammatory response. And this inflammation can induce the tauopathy to start developing in the neurons. And it's the tauopathy that causes the neuron death. We have mouse models that have only tauopathy, and their brains shrink aggressively. To the point that they can sometimes be 1/2 of the normal size of a normal mouse. I'm amazed that a mouse can survive with half of its brain. But this is showing us that tau is really what is causing this neuron death. The genetics of Alzheimer's, as I mentioned, has been very informative. And it's really pointed us in a couple of directions. And the first one gave us the impression that it's this amyloid that is kind of the initiating factor in the disease. Down syndrome is caused by having an extra copy of chromosome number 21. And Down syndrome individuals do have cognitive impairments in their youth, but in addition to that, when they reach their 40s and their 50s, they often will show a dementia syndrome in addition to the cognitive problems they've had up until that time. They have an extra copy of the amyloid precursor protein, the protein that makes the amyloid, because it's located on chromosome 21. All Down syndrome individuals in their 40s and 50s at autopsies will have plaque and tangle pathology, just like an Alzheimer patient does. There are also these rather rare familial cases of Alzheimer's where there's mutations in the amyloid precursor protein, or interestingly, the proteases that cleave amyloid out of the amyloid precursor protein. And these proteases have some raggedness to their activity, and they can make either a 40 or a 42 amino acid amyloid peptide. The 42 amino acid peptide aggregates more quickly. And many times what these mutations are doing is not changing the amount that's made, but the length of the amyloid peptide. And that's enough to condemn you to developing the disease in your 40s and 50s. There are a number of genes which increase risk. Some of these are very close to being dominant. If you get 2 copies of the apolipoprotein E4 variant, there's 2, 3, and 4 normally in our population. If you have 2 copies of E4, you have a 10-fold increased risk of developing the disease. It's a massive increase in risk. We've also found recently, though, as I mentioned, that a number of proteins involved in innate immunity. And so while their numbers are small, the frequency of these mutations is rather low, their impacts can be just as high as that of ApoE4. And so this has really been helpful to us in understanding the role of inflammation in the disease. And finally, we know there are some individuals that don't have any known risk factors for the disease based on their biology. But there's lots of discussion about, this study shows that, taking this vitamin reduces your risk, et cetera, et cetera. But the things that have come through consistently have been in particular head trauma. We're very concerned about what's called chronic traumatic encephalopathy that results in a tau deposition in the brain. It evidently also initially has amyloid deposition in the brain, but the amyloid gets cleared out, but the tau remains. And this is how head trauma, repetitive head trauma, can increase risk. Diabetes appears to be associated with increased risk of dementia. People who have cardiovascular disease also have increased risk of dementia. Hypertension in particular increases your risk for dementia. There are some things that decrease your risk, interestingly. Education has a profound effect. In fact, people who have less than a high school education compared to people who have a postgraduate degree have a threefold increased risk of developing dementia. It's actually the same as having a single copy of the apolipoprotein E4 allele. And finally, one which again focuses on this idea of inflammation. People who have one of various forms of arthritis have a decreased risk of developing dementia. And for many years, we thought, well, it's because they're taking these anti-inflammatory medications. But when we did the randomized, double-blind, placebo-controlled trials, there was zero effect of those medications. So instead, what it may be telling us is there's something about the innate immune system in people who develop arthritis that protects them from developing dementia. And one possibility is their immune system reacts early to the amyloid deposits, clears them out, and the downstream effects of amyloid don't occur. We don't know that. That's just an idea. So for a long time, in order to give you a diagnosis of Alzheimer's, we had to take your brain out, section it, stain it for amyloid and tau, and put it under a microscope. And most people preferred we not do that when they were still alive. But recently, we've now been able to develop PET ligands that can detect amyloid, and I'm not going to talk about it, but now also tau in the brain of a living human being. What these ligands do is they essentially stick to the amyloid in the brain. You inject them, initially they go everywhere, the excess washes out, and you then take a picture with a PET camera to see where the chemical is. And if the chemical's there, you get a higher signal. And so what we found was that people who were normal had very little amyloid in their brains, and people who had Alzheimer's had a lot of amyloid in their brains. And in correlation with that, another way that we do PET scans is to use a sugar that's got the same PET ligand or PET signal associated with it. And what you find is that people who are normal, their brains are taking up lots of sugar out of the blood. And that's what you see on this lower panel. Whereas those who have dementia have very little sugar uptake. Unfortunately, all kinds of dementia will cause this decrease in sugar uptake. But only in the Alzheimer cases do we see the increase in the amyloid. So this is now permitting us to identify people who really have Alzheimer's instead of just some other form of dementia. Much better than simply a clinical diagnosis and without having to do a pathological analysis. So when we started doing these PET scans, we found that about a third of the normal people had amyloid in their brain. And at first we thought it was an artifact, but then what we saw is over the next 5 years or so, these were the people who are more likely to develop Alzheimer pathology and to develop essentially dementia. It put them at a much higher risk of developing dementia. And that's how we now know that it's 10 to 20 years before the onset of symptoms that amyloid is building up. So this is what we think is happening. This is the line where you first notice any symptom at all, and that's almost always done retrospectively. This is the line where you get a diagnosis of dementia, which means you no longer can live on your own independently in a safe and effective manner. Amyloid is building up, and by the time you show first symptoms, you've got all the amyloid you're going to have. Tau is much delayed, and by the time you start to show symptoms, you've got half the tau. That all is associated later with an atrophy of the brain, a loss of brain mass, finally memory is affected, and then activities of daily living. And so this whole span is probably about 30 years that we're looking at here. So recently, there have been two medications that are based upon the idea of trying to remove amyloid from the brain. It's called immunotherapy. I actually published a paper on immunotherapy in Nature in 2000. So it's been 20 years since we published our first paper showing that immunotherapy could reverse memory loss in mice. And now we're starting to find drugs that actually are starting to do the same thing in humans. This is a drug called aducanumab. It's an antibody against the amyloid peptide. And what we're seeing are these PET scans that I just showed you. So this is the baseline and this is the placebo. You see there's no change. A low dose, you see some reduction. A middle dose, you see more reduction. And essentially the highest dose, you almost make people amyloid negative. They essentially don't have detectable amyloid in their brains. When we look at the reduction in amyloid, you can see the time-dependent reduction relative to the placebo. And if we look at cognitive decline, by the time you start to really getting rid of it, it starts to level off. It starts to flatten out. This is a second antibody, and it does much the same thing as the first one. And here you see a dose-dependent reduction in the amyloid in the brain as measured by the PET scan. And when you look at the rate of decline cognitively, at the highest dose at least, there's a 50% slowing of cognitive decline compared to the placebo. So this is very encouraging data. These drugs are still under further development, but they're being considered by the FDA for approval. This one's in phase 3. The other one completed phase 3 and is being evaluated by the FDA. So, but what this is showing you is we're now closing in on having some medications that can actually modify the disease process. So we think over the next 5 years that we're going to have a situation where people in their early 60s can go in and have a PET scan or possibly even a blood test to determine if they're developing amyloid in their brain. If you had a positive family history or a known genetic risk, you might want to do this a little earlier. People who show these amyloid deposits will be given options of using amyloid-lowering medications, and there's a number of avenues that are using various approaches to do this. We might even be able to have a vaccine against amyloid that could do this. But they'll have to, balance the risks and benefits because no medications are without risk. It's not going to be a slam dunk, but people are going to have the option of making those decisions. We think this will either delay the onset by years or perhaps even prevent the disease entirely. So we're hoping that by 2025, we can really start to make Alzheimer's a memory. And that's actually the slogan of the Alzheimer's Alliance. I'm just going to briefly touch on some clinical work we're doing. We have a community-based memory screening program. We're training volunteers to conduct free memory screening analyses of residents in their communities. This is downtown Grand Rapids. All of these stars are locations where we're doing memory screening in YMCAs, elder law attorney offices, residential care facilities, senior centers, et cetera. We're also running a clinical trial looking at cognitive training to see if this can in fact slow the progression of Alzheimer's disease. We're presently on pause with this, which has been very frustrating for us. We can't see anybody because of COVID. We're hoping to get this started up again soon, but we often need participants to join in these studies. And then we want to try and develop pharmaceutical company-sponsored clinical trials. And to do that, we need volunteers to come in and act as mock patients, mock participants. So our staff can demonstrate that they have proficiency in giving the instruments that we use in order to test these medications. And so these phone numbers, if you call them, we can easily help you if you wish to participate in mostly people in the Grand Rapids area here. But we have a variety of opportunities for people to volunteer to help move forward with this program that we're developing. Finally, these are the folks that are working in the lab. These are some of the other Alliance members here in Grand Rapids. This is our research support. And again, making Alzheimer's a memory is our final goal. Thank you very much.
Thank you, Dr. Morgan, for that wonderful talk. I'm an arthritis person, so I was happy to hear that maybe all my joint pain is helping me in some fashion. So that was good news to hear.
That can be, it surprises people, but it's a relatively consistent observation that, particularly things like rheumatoid arthritis tend to be negatively associated with risk for developing dementia.
That is really interesting. With your community screening and things, is that only in Grand Rapids, or is there other locations like here in East Lansing or the Detroit area if people were interested?
Well, it's interesting. It's only in Grand Rapids right now. I actually wrote a grant last year to try and take it statewide, and it didn't get funded. At least at this point in time, we don't have plans to expand beyond that, but it is a very scalable program. I actually started this down in Florida in the retirement centers, where we would actually go into these, in Florida, you've got all these people over the age of 55 that decided to all live together. So it's all the people at risk of Alzheimer's now are very close to each other, and they all have a rec center in the middle. And so we could train their retired nurses to give this Montreal cognitive assessment, and people can just, walk down to their rec center and get a free memory screening. We took their information and put it into a registry. And so when clinical trials came about, that might be appropriate, we could identify potential participants in those clinical trials. We also developed a mobile clinical trial unit. It was a trailer that had everything we needed to do the clinical trial. So we could take our mobile unit, park it in their rec center parking lot. They could come in their golf carts and their flip-flops and be part of a research study.
That's fantastic.
That won't work in Michigan, however, because we have this thing called winter.
Yes, that is true. Although there are some larger, like assisted living places that do have the rec rooms and things. So I would think maybe there's a way to make that happen.
Well, and we are using those for many of our screenings here in Grand Rapids.
We did have a question. Samantha wanted to know if there was an email that she can reach out and get more information about this research program or laboratory.
So I think if what you wanted to learn about was the clinical part of it, it would be you just go to the Alzheimer's Alliance website and you can find the email link for the various programs that we have there. I have a very simple email. It is scientist.dave@gmail.com. So I can talk to her about anything to go on at the laboratory.
Okay. And we can also put up it's alzheimersalliance.msu.edu as your website. You can put that up as well.
That is correct.
Awesome. Perfect. So with the potential immunotherapies coming, if you have a family member that you see some changes, can they currently go ask their doctor to get a PET scan to see what's going on, or is that still at the research level?
So right now you can do that, but the real problem is that Medicare is not reimbursing for it at this point in time, and it costs about $5,000.
Right.
So you can do it, but most people don't, because the cost is for most of them prohibitive. The one thing you can do if you can find a clinical trial, however, is that almost all clinical trials of Alzheimer medications are now giving a PET scan before they enroll you in the study. I skipped over a couple of slides there. But in those slides, we found out that about 20 to 25% of people diagnosed by the best physicians, the ones running the clinical trials, as Alzheimer's disease did not have amyloid in their brain. 20 to 25%. So you're treating them with an anti-amyloid drug and they don't even have any amyloid. It's kind of doomed to failure, right?
Right,
right. So we now largely are always scanning people, and if you participate in a clinical study or are willing to do so, you can get a free amyloid imaging scan and find out if you have amyloid or not. You can then say, well, I've decided I don't want to go into the study. There's nothing that we can do to block you from doing something like that. Unfortunately, right now, I don't know of any studies enrolling in East Lansing. Andrea Bazzoki was doing studies in East Lansing. She's recently left MSU. And I don't know that, I think that the people who were enrolled in the studies already are still ongoing, but I don't think they're enrolling anybody new. I'd have to investigate that more, but yes, we're trying to convince CMS CMS is the organization that approves reimbursements for Medicare and Medicaid. And they recently ran a study called IDEAS, where they did 18,000, they took in people, had them do a diagnosis with a memory specialist physician, and the physician developed a treatment plan. They subsequently gave them a PET scan. And then they came back 30 days later and they asked the physician, okay, now we've told you what the PET scan was. Did that have any impact on your treatment? 65% of the time, it changed the treatment plan. It was twice as much as they expected when they designed the study. So it obviously is having an impact. And I think what's going to really the problem is this is a very expensive procedure. And there's several developments that I think we're very close to having. And one of them is measuring the amyloid in the blood in certain types of circumstances using certain techniques. We can predict who is likely to have positive amyloid by PET scan about 85 to 90% of the time. And so you'll probably have the blood test done first. And if that shows a very high likelihood, then we would go ahead and do the PET scan, the much more expensive but definitive test. But I also think that once we get a drug that actually is approved by the FDA, that is working on amyloid, that would then justify doing an amyloid PET scan to find out who should get the drug and who shouldn't. It's kind of like, doing the genetic analysis of a breast tumor to determine what medications you should get. You need to have the diagnostics along with the therapeutics.
Right. Well, we hope that with the increased funding, and we hope that they also increase funding beyond what it is now, since it matches cancer and cardiovascular disease, that we'll see some of these things, as you say, before 2025 and help out families out there that have loved ones that are affected by this.
So an interesting little aside about the lobbying that I've done to try and increase funding. I'm the lead representative of an organization called Researchers Against Alzheimer's. There's about 500 scientists working in this area that have joined this organization, and it's a very loosely knit type of organization. But it's part of a larger group called Us Against Alzheimer's, which has thousands of people and has a single goal in mind, which is to increase funding for research in Alzheimer's disease. And in that capacity, I have gone to Capitol Hill probably about a dozen times over the last 8 years or so. And recently, last fall, we had a cocktail reception in the House office building. And there were 7 members of Congress set up to come and talk. In prior meetings, they would have 1 or 2 of them would actually come. The rest of them would send staff members to go and give this. This time, all 7 came themselves to talk to us. And the most amazing thing is they were feeding back to us the talking points that we've been giving them for the last 8 years. They actually heard us. I was amazed.
Oh, that's awesome. That's what we need to have happen. Well, we are out of time, sad to say, but we want to thank you for being part of the MSU Science Festival Science After Dark program. We do have another talk coming on in about 15 minutes on opiate awareness. But if anybody has any questions, they can put them in our comment box and we will get answers for you. So thank you again, Dr. Morgan, for joining us.
You're very welcome. Thank you for inviting me.
You're welcome. All right. Take care.
Bye.